So these will be the last slides of the sensory that we didn't get to in class before all this came to fruition. But again, I hope you all are staying safe. But we saw the signaling of the photoreceptors in the eye, in the retina. And we've seen we have rods and we have cones, and in the cones are going to give us our color vision. And you can see we got three different colors, or three different cones. Basically we got the blue or S cone. Okay, the blue S is for because it is shorter wavelength, and then we got the green, which is the M cone. Okay, for medium, since it is the pigments pick up medium wavelengths, and then red or L cone for long because it picks up those pigments of those cone pick up the longer wavelengths. Okay, so we saw the signaling in the rods, in the rod photoreceptors. Okay, they had rhodopsin. In this case, there are three different color photoopsins, and they pick up those different color wavelengths, those photoopsins. Okay, but signaling is going to be very similar to the rhodopsin that we saw from the slides in earlier with the photoreceptor signaling. Okay. And so this is how we get our colors. We get a combination blue, green, and red. So depending on what light is being reflected back to the eye, if something's black, it is absorbing. None of these lights are being reflected back. So then these cones aren't being activated, and the brain can put an image, hey, I'm not getting much signal from all three cones, and I'm interpreted as black. So that's what the brain's going to interpret when it gets the information gets back to that primary visual cortex. Okay. Let's say if it's what's getting reflected, say the color of the shirt is like a cyan color, okay, even like a lighter greenish blue than this, the cyan that you see in the printers, then you would get in some firing of this, and that color also falls within the M spectrum, so you get some firing of the cones with the green. So your brain is going to get a combination of some blue, some green, and then the brain can put the color together as somewhere in that cyan color, that greenish blue, kind of like a turquoise almost. Okay, so that's how it operates. Depending on what signals are being sent to the brain, which cones are being activated, because of what light's being reflected and refracted into the eye, determines on what color we see, and it's because it is coming from these trichromatic cones that we have. So let's look at where those cones are located. We've seen your eye, because a high density of photoreceptors, specifically the cones, are going to be in that little dot right here, that fovea centralis. Okay, the fovea centralis resides in this larger area that has substantially high density of cones. Okay, again, the fovea centralis is the highest density, but the macula lutea, this larger region, is basically where the light is being focused onto the retina. Okay. And so highly pigmented area of the retina, but again, the sharpest vision is going to be at the fovea centralis. So that's why your eye muscles move your eyeballs, so the light is reflecting back into this area. And many people have probably heard of macular degeneration. That's that basically macula lutea. That region of the retina is starting to degenerate. And remember, your eye, no matter even if your macula is degenerating, your eye is trained to focus on, this should be focused near the fovea centralis. And so that's what your eye is going to do. But in this case, that area around the fovea centralis, the macula lutea, is going to be degenerating. So people see like a blob, okay? Or they'll see blurry, okay? You can see maybe off to the sides a little bit better because the macula is not degenerated there, but because this is where the eye is going to focus, whatever's in the focal point will be blurry, and in this case you'd have this almost like a black hole if you were staring at this and had macular degeneration. Okay, so let's look at what's happening at this fovea centralis. Why is there so much, why can we get so much acuity? All right, why is the greatest acuity at this fovea centralis? And you can see the cones here. Okay, the cones are going essentially 1-to-1, or there's not a lot of convergence. So what light hits here is going to stimulate one neuron going to the brain. So basically the visual field is just a small area, where if you have more convergence, you'd have a larger, say, area. Okay, so you can get fine-tuning with this low convergence coming up to the bipolar cells. Also, you can see there is this little indentation, and that is because, remember, the light is coming through. It has to come through, okay, this neural layer here for most part. But in the fovea centralis, because these neurons get parted, the light that's coming in here has a clearer shot at these photoreceptors down here. Okay, so the amount of very little convergence, okay, we don't have 3, 4, 5 cones converging onto one bipolar, okay, typically 1-to-1. Here it's 2-to-1, but typically it's 1-to-1. And then the other factor is you have this light is able to, because of this parting of the neurons here, you have the ability to pick up that light much greater. And so once we stimulate, which way does the signal go? It goes from the photoreceptor to the bipolar to the ganglion cells. And the ganglion cells are going to make, right, here are the ganglion cells making the optic nerve, okay, giving us that optic disc or blind spot back there. And so once we have that signal coming through the optic nerve, we'll be going. Let's look at the acuity. This is just a little blow up. This is looking at the convergence. If you can see with the rods here, what do you have? You have, here's the ganglion that's going to go and bring the message to the brain. What is it stimulated by? It's by this whole, all of these would stimulate the same ganglion. So you can't, any light that's here, the brain's going to pick it up as just one signal. Whereas with no convergence, what you see in the fovea, you see 1-to-1. So this little region is only going to stimulate this, and this little region of light hitting here is only going to stimulate another ganglion neuron, and this little region here where light would hit would stimulate another. So you can get finer, crisper vision because you are getting more signals. There is less area being picked up. See this small area compared to all of this area is just activating one. The brain is going to get the signal from one ganglion cell. Okay, so the receptive field is much smaller for here, where it is much greater when we have convergence. Okay, so again, light comes in, we go from the photoreceptors up to the bipolar cells and up to the ganglion, and then this is going to make the tracks to the optic nerve. So once we get that information traveling through the optic nerve here and then to the optic chiasma, where we cross over, and the optic tracks, eventually we're going to end up at the primary visual cortex. Some of the information will go to the lateral geniculate nucleus of the thalamus. Okay, but the way this runs is a little bit different than, say, the cutaneous. If we saw the cutaneous, it was contralateral. Touch receptors on my right side of the body, that information is going to travel and go to the left side of my brain. For vision, it's the field of vision. So the left field of vision, you can see it's going to hit, where's it going to hit? It's going to hit parts of the left eye, but also parts of the right eye. Okay, the lateral side of the right eye, but the medial side of the left eye. But the left field of vision information is all going to cross over and go to the right side of the brain. And then the right field of vision, again, parts going to go to the right eye, and other parts are going to go to the left eye from the left or right field of vision, but all of the right field of vision is going to end up on the left side of the brain. Okay? So it's not the actual physical part of the body, but it's the field of vision. Right or left is going to go to the different side of the brain. And we saw the two pathways. We have the geniculate nucleus. Okay? That is the what pathway? That is going to tell you what you're looking at. Okay, or the tactile. And back here, it's going to give you the where. Okay, where is the information coming? Where in the field of vision is it? And so that finishes it up with the eye and with the sensory here.